Hello, good morning. Um, I'm Ali Udanteni Abdullahi, and going to be in charge of teaching this course. I think the course code is BCHM242 for the nursing students, right? From DLC. All right. Summarily, I'm sure this is not your first rodeo, right? Usually, most of these courses have three modules. Each module has 4 sessions, right? 4 study sessions. Okay, so we're going to start with module 1, which uh, covers metabolism, basically. I mean, the whole course is metabolism, but the carbohydrate metabolism, more or less, is covered in module 1. Fatty acid oxidation, glycolysis 2. TCA cycle, that's the citric acid cycle, and fatty acid oxidation. Well, from a biochemical standpoint, we we won't approach these subheadings like this, but it's okay since this is meant for nursing students. So I think it still passes the message across when handled adequately. Anyway, I think we can get started. This is your grading criteria. You're also familiar with the website, so I'll be dropping instructions on assignments, individual assignments, group assignments, discussion, and quizzes, because that will be uh, your continuous assessment. That will form part of your continuous assessment, basically, right? All right, so glycolysis and the glycolytic pathway, right? So if you come to your study session 1, okay, glycolysis 1. So what's glycolysis? Glycolysis, as you can see, is a sequence of reactions that converts glucose into pyruvate. Glucose is a 6-carbon sugar. We are familiar with sugars, right? We eat a lot of cereals here in Nigeria. The tuwo, your rice, your maize, those are carbohydrates, right? So we consume a lot of sugars, and of course, our body will have to be equipped with enzyme machineries to catabolize or break down those sugars into useful energy, which in the cell is called ATP. That's the energy currency of the cell, ATP, adenosine triphosphate. So glucose is converted into pyruvate to the production of ATP. That's basically what happens with most of these pathways. That's the purpose of metabolism. So in aerobic organisms that carry out their metabolism in the presence of oxygen, right? Like humans, we inhale air, right? We inhale oxygen and we exhale carbon dioxide. So in aerobic organisms like us, right? Glycolysis is the prelude to TCA cycle. Now, this is just the basic introduction. I'm going to familiarize you with the pathways, So you see, uh, you get a picturesque representation of what's actually happening instead of just reading and trying to memorize without understanding what's going on. From the glycolytic pathway, we go into the citric acid cycle, and the products of citric acid cycle go into the electron transport chain, where we end up producing the energy currency that's needed to boost the performance of your cells, tissues, and organs, right? So if you read this, you realize that under aerobic conditions, pyruvate enter the mitochondria. Of course, you are familiar with the mitochondria, right? As nurses, you do anatomy, physiology courses, so you should be familiar with the cell. The mitochondria is called the powerhouse of the cell. So in aerobic organisms, the catabolism or breakdown of carbohydrates, sugars, lead to the entry of intermediates of this reaction into the mitochondria, because the mitochondria is the powerhouse of the cell. And these intermediates, when they enter the mitochondria, they are completely oxidized to carbon dioxide and water. So if the supply of oxygen is not sufficient, the body will have to manage this an optimization that will have to go on. In the absence of oxygen, is the process supposed to stop? No, it shouldn't. So the body will, like the muscles, for example, say in a sporting condition, when you're running, if you're an athlete, you run, and you don't have adequate oxygen. The pyruvate from the glycolytic pathway is converted to lactate. That's lactic acid, basically. So it's part of what causes muscle pull. As nurses, you should know, right? Muscle pull. So basically, this is just the basic introduction as to what we'll be getting into in this course, right? So now we've mentioned glycolysis, TCA cycle, and the electron transport chain. Now, in biochemistry, you don't just stop at the periphery. You don't stay on the surface. We like to go deep, right? We like to go dive inside, see what's actually happening. And that's why we have to study the glycolytic pathway in itself. It's a pathway where glucose is converted to pyruvate. So the series of reactions that occur, the enzymes that are involved, we have to know those enzymes and those intermediaries they produce, right? So basically, I think this chart here summarizes everything. This is the glycolytic pathway. Okay? This is the glycolytic pathway, glycolysis, and glycolytic enzymes. Now, what are enzymes, right? Of course, you should know what enzymes are. They are biological catalysts that speed up the rate of biochemical reactions in the cell. So your glucose here is in a chemistry term, they will call it a reactant, but in biochemistry, it's a substrate. You feed this substrate to the enzyme, and the enzyme expedites the production of your products. This is what's happening here. These are your enzymes. One here is hexokinase. Glucose is converted to glucose 6-phosphate by hexokinase. You need an energy investment into this reaction, energy in form of ATP, which donates the phosphate that is added to glucose at the 6th position. And then your ADP is produced. This ADP is one phosphate short. It started with adenosine triphosphate, 3-phosphate. Now we have adenosine diphosphate 2, right? So there's an energy investment here to produce this, and the enzyme that catalyzes, that speeds up this production of glucose 6-phosphate is hexokinase. So now from glucose 6-phosphate, we produce glucose fructose 6-phosphate. An enzyme is also involved. This is the name of the enzyme, phosphoglucose isomerase. So basically, this is what happens as you go. Now your fructose 6-phosphate needs to now be converted to fructose 1,6-bisphosphate. Bisphosphate means you have 2 phosphate groups. So this phosphate group has to come from somewhere, right? It doesn't just come out of a vacuum or something. So you have another energy investment of ATP here into this reaction. So your cells, your body has invested energy into producing these intermediates of the glycolytic pathway. Now, this is your initial step of the pathway. Conversion from fructose 6-phosphate to fructose 1,6-bisphosphate is catalyzed by phosphofructokinase 1. Okay? Now, your fructose 1,6-bisphosphate has to now be lysed into 2 compounds. Aldolase, it's a lyase. Lysis breaks down this compound into 2 components. First component is your glyceraldehyde 3-phosphate. The second component is your dihydroxyacetone phosphate. All right. Now these 2 compounds can interconvert into one another. I don't know if that's the right word. If you look at this arrow here, it tells you that this reaction is reversible. Dihydroxyacetone phosphate can be converted to glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate can be converted to dihydroxyacetone phosphate, right? The enzyme that can do that is triose phosphate isomerase, right? And now also looking at all these reactions, these arrows that show forward and reverse, it means the reaction here can be reversed, right? The reaction here cannot be reversed easily. You see, this is irreversible. So now I'll have to explain the implication of these irreversible reactions. The one for hexokinase, the one for phosphofructokinase 1, and the last one here, pyruvate kinase. You see the arrows, it's just an arrow going in one direction. It's not reversible. All right. Now going back to your lecture note, here we are told that the glycolytic pathway has a dual role. It degrades glucose to generate ATP. So the purpose of this pathway is to produce energy, right? Well, if you look at the initial phase of the pathway, we've not produced energy, but we've invested energy already. Two reactions where we had to invest ATP to phosphorylate a product, right? I mean, you still remember, if you look at this is an energy investment. This arrow here, this is an energy investment. It's going in. ADP is going out. It's not ATP that's coming out, but here ATP is coming out. Now we are producing energy here. We are producing energy here. Now, it means for you to produce ATP downstream, there's a need to invest energy in the initial phase. Basically, that's what's happened. It's just like making money. If you want to make money, sometimes you need to use money to make money. And I think that's a layman example, but it makes sense. Okay, so it degrades glucose to generate ATP. It provides building blocks for synthetic reactions. We'll key into this in a minute. And then the rate of conversion of glucose into pyruvate is regulated to meet these two major cellular needs. So, well, this is mentioned two roles of the glycolytic pathway. I think we just read them, right? We should know it degrades glucose to generate ATP, provides building blocks for synthetic reactions. So the intermediates of glycolysis can go into other pathways, synthetic biosynthetic pathways in the cell. The glycolytic glycolysis breakdown of glucose. It's the breakdown, metabolism. It's either you break down or you build. So the breakdown is catabolism. The production biosynthesis is anabolism. So basically, that's what happens. So the two types of metabolisms we have are catabolism, that's breakdown, just like in glycolysis, or anabolism, that's the biosynthesis, like ketogenesis, for example. Okay. Or glycogenesis, that's another example of biosynthesis. Gluconeogenesis, that's another example. All right. So the stages of glycolytic pathway. So you have the first stage, which I think I've already mentioned already. Yeah. I don't need to go into this again because you've mentioned that. Okay, so this point is very important. This point here. Okay, the reaction catalyzed by phosphofructokinase, an allosteric enzyme. I wanted to mention this point. Why? What's an allosteric enzyme? So this here, this reaction is not reversible. Conversion of fructose 6-phosphate to fructose 1,6 bisphosphate it's not reversible, and it is catalyzed by phosphofructokinase, which they termed an allosteric enzyme. So allosteric enzymes are basically, what are enzymes? We've defined an enzyme, but at least we should understand what enzymes are, right? So this is the enzyme. An enzyme is a biological catalyst. This is a substrate. Let's call this glucose. The substrate is glucose. Glucose will bind to the catalytic side of the enzyme. This is where glucose is supposed to bind here. Now, this site here is what we call an allosteric site, not the active side of the enzyme, not the catalytic. The catalytic site is also the active site, but an allosteric site is a site that is not meant for the substrate. But this site is a site that can be explored when you want to regulate the activity of this enzyme. Say the enzyme has produced enough product. A compound, an inhibitor, can bind to this site to prevent the active site from working. So basically, that's what an allosteric site is. So the inhibitor binds to the allosteric site of the enzyme substrate complex. For example, this is it. This binding of the inhibitor, right, to this will prevent the enzyme from converting more. So it's a regulatory mechanism. Of course, you want glucose to be broken down so you get energy, but too much of that is also a problem. So there's a check and balance in your system, right? So that's what that's all about. All right. So that's why phosphofructokinase 1 is an important enzyme. It's a regulatory enzyme in glycolysis. They call it the rate limiting step. After this step is a point of no return in glycolysis. The moment you pass this step, then glycolysis has to occur. So the three checkpoints in glycolysis are this, the first one here, this, and the last one. And you see the reactions are irreversible. But this phosphofructokinase 1 is called the rate limiting step. You know, that's a point of no return. Okay. Let's go back to your lecture note. Okay. So we don't deviate too much. Now, the pace of glycolysis is critically dependent on the level of this enzyme. You see, if you understand this statement, the level of this enzyme will guide the pace of glycolysis, whether it will happen at a fast or slow pace, or whether it will even happen at all. Its catalytic activity is controlled by ATP and other metabolites. So technically, this statement is saying when enough ATP is produced from glycolysis, ATP can become an inhibitor of the enzyme. It's this kind of inhibition where the product inhibits the enzyme is called feedback inhibition. When you have enough ATP, the reason why glycolysis will occur is because there's an energy deficit in the cell. Your body needs energy. But when the glycolysis has produced enough ATP, the pathway has to slow down, right? So what better to slow down the pathway than the product of the pathway? And since the enzyme is an allosteric enzyme, it has a binding site, that a regulatory site where something can bind to prevent it from continuously convert a substrate to a product. Then ATP should serve as a good inhibitor in that regard, right? So its catalytic activity is controlled or regulated by ATP and other metabolites. So I hope you understand. I really hope you understand. Okay, so the second stage of glycolysis. Now, look at this stage 2. The first stage, okay? The first stage is here from glucose to this stage here. Glucose to fructose 1,6-phosphate before aldolase cleaves this compound fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde phosphate. Now, this is where the energy investment occurred. We didn't produce ATP here, we invested ATP, which is the energy currency of the cell. Now, this is the preparatory phase of glycolysis. All right, this is the preparatory phase, and that's the first stage of glycolysis. The second stage will now be where the production of ATP will occur. All right, this is it. This is it. Now, after aldolase, this enzyme cleaves fructose 1,6-bisphosphate into these two compounds, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. You have one molecule of glyceraldehyde 3-phosphate, one molecule of dihydroxyacetone phosphate. But dihydroxyacetone phosphate is converted to this because the reaction is interchangeable. It can go forward and reverse. So we need glyceraldehyde 3-phosphate for glycolysis. We don't need this right now. So this dihydroxyacetone phosphate is converted to this, and that's why you have these two here. It means after this was converted to glyceraldehyde 3-phosphate, we now have 2 molecules of glyceraldehyde 3-phosphate. All right. Now, glyceraldehyde 3-phosphate dehydrogenase is now going to convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This is an oxidation reaction, and we need a coenzyme. Coenzyme are organic compounds in the cell that are needed by enzyme to catalyze their reactions. So this coenzyme is needed by this enzyme, NAD+. We have NAD, we have FAD. Those are examples of coenzymes, all right? Now, this coenzyme plus 2 inorganic phosphates were fed into this reaction in addition to the glyceraldehyde 3-phosphate. We have 2 molecules of this coenzyme and this because we have 2 molecules of glyceraldehyde 3-phosphate. And this ended up producing 2 molecules of NADH and protons, right? Okay, now these NADH have their function in the electron transport chain. They can be converted to energy there. Well, let's focus on what we are doing here now, right? Let's not run faster than our legs. Now, 1,3-bisphosphoglycerate, 2 molecules now of it, because we are working with 2 molecules now. We know there's a balance in chemistry. There's a balance, you have to maintain balance. You're working with 2 molecules, 2 balances 2. If you have 2 inputs, you should get 2 outputs, right? Okay, so 1,3-bisphosphoglycerate is now converted to 3-phosphoglycerate, enzyme 7-phosphoglycerate kinase will do this. And you have 2 ATP, 2 ADP is inserted here, and you produce 2 ATP. Now, this is where the production of energy currency comes in, right? We have energy currency produced, okay? Now, we have 2 ATP. Now, 3-phosphoglycerate is now converted to 2-phosphoglycerate by phosphoglycerate mutase, right? It's just the change of the phosphate group, position of the phosphate group changed from position 3 to position 2. That's why you have this. And a mutase does that. And then 2-phosphoglycerate, 2 molecules of it is converted to phosphoenol pyruvate. This is a dehydration reaction. Enolase will dehydrate this. And you have okay, you have the production of phosphoenol pyruvate, and then pyruvate kinase will phosphorylate. Now, the purpose of a kinase is phosphorylation. That's why you have this phosphoenol pyruvate, you have your pyruvate, and that phosphorylation, the phosphate group, will lead to the production of ATP. Okay. Now, looking at this now, you have 2 ATP here plus 2 ATP here. The glycolytic pathway has produced 4 ATP. But why do they have 2 ATP here? It's because we invested 2 ATP in the beginning. ATP went in, it didn't come out. Another ATP here went in. So 1 plus 1 is 2. 2 plus 2 here, 4. Production, right? We've generated 2 ATPs. So 4 minus 2 will give you 2. So the glycolytic pathway gives you 2 ATP. And it also gives you 2 NADH. This is important for the electron transport chain. 1 NADH, if 1 NADH enters the electron transport chain, it produces 2.5 molecules of ATP. While FADH, when they enter the electron transport chain, they produce 1.5. You should take these figures very importantly. You should write them down. 1 NADH, 2.5 molecules of ATP, 1 FADH2, which we will encounter in the citric acid cycle, produce 1.5 molecules of ATP, okay? So, and then we have 2 pyruvate, of course. We're working with 2 molecules from here. So, that's the glycolytic pathway in a nutshell, okay? So let's go back to your notes. I think basically everything that's here, I've already mentioned it, but you can read more, right? They broke down this into 3 stages. I think we are still saying the same thing, okay? This is the glycolytic pathway. You take your time and read. You might be, the pathway might be new to you. Of course, you know glycolysis, but knowing it from a biochemical standpoint is something else entirely. So now looking at the 10 reactions of glycolysis result in a net gain of dash ATP and dash NADH22, right? I just finished presenting it here, okay? Right? You can take your time, follow the reaction and see for yourself. Okay, so tutor marked assignments, individual or group. So I think I'm going to make it an individual assignment. Of course, it's not very difficult. I want you to list the classes of enzymes involved in glycolysis and the reactions they catalyze. This shouldn't be difficult. So I hope you can submit these assignments to me, hopefully by Wednesday, please. I'll post it in the group. I'll post the assignment in the group. Then I'll be expecting you to please, send it back to me. I think I'll do that. All right. And then you can read the conclusion. Glycolysis is the key pathway that other pathways depend on, either in the provision of energy or as a building block for the initiation of other pathways in metabolism. Okay. In this study session, let's read this again. Glycolysis is the key pathway that other pathways depend on. So this pathway is like a central pathway in metabolism. Other pathways depend on it. They depend on it for provision of energy, okay? They depend on it for provision of energy or for building block for initiation of pathways in metabolism. It's either production of energy, catabolism, or for biosynthesis of molecules, that's anabolism. Okay, basically this statement just described metabolism. I apologize for that. I had a network problem. Let me share my screen again. Okay, so self-assessments. As directed in the laboratory, please answer the following questions. Explain the concept of glycolysis. Okay, stage 2 importance of the glycolytic pathway, and then enumerate the reactions which make up the pathway in stage 3 and the enzymes which catalyze these reactions. So when you read your lecture note, you should be able to tell the stage 3, right? I think in your lecture note, in this stage, 3 enzymes are involved. Generation of pyruvate, 3-phosphoglycerate is converted to. So basically from this reaction, 3-phosphoglycerate here to the end is your stage 3. Okay? While your stage 2 is the production, the splitting of fructose 1,6-bisphosphate to the production of 3-phosphoglycerate. All right, so still not a big deal, really. But yeah, so I think you should be able to do this. I'm going to make sure I tell you, I inform you, I pass information to the platform. I'll send it officially as an assignment to the group. So it will be registered in the website, okay? You read the reactions which make up the pathway in stage 3, and okay, this is not a big deal also. So I think if you understand, this is very straightforward. And you have this recording, you can always replay if you don't understand any points. At the end of this module, we are going to have a studio session where we interact. And if you have any questions, ask them, okay? All right. So I'll have another record session for study session 2, hopefully. It shouldn't be here, okay? Thank you.